the CEO of the Global Sepsis Alliance, I'm honored to moderate this important session with a distinguished panel of speakers. In the next 80 minutes, we will discuss the immunization agenda for sepsis. We will speak about the importance of existing routine vaccination for women, children, older adults, and other high-risk groups. And we shall also explore the research agenda for new vaccines against the most common pathogens leading to sepsis. We will explore the context of both high-income and low and middle-income countries. And I'm delighted to announce that we have already exceeded 10,500 registrations for the current online Congress from across 150 plus countries. And we look forward to active engagement in the Q&A session with the audience. Please send your comments and questions to the speakers in the chat box under the live stream. Without further ado, let me introduce our first speaker. It's my absolute delight to present to you Professor Stefan Kaufmann, globally acknowledged scholar in immunology. Stefan is the founder and director emeritus at Max Planck Institute for Infection Biology in Berlin. He has held prominent academic positions at the Charité University Hospital, Texas and Marcel Universities. He's the former president of German Society for Immunology, European Federation, and International Union of Immunological Societies. Our speaker is the developer of a tuberculosis vaccine that is in phase 3 clinical efficacy trial. And Professor Kaufmann is the recipient of numerous awards and the author of over 900 publications. Stefan Kaufmann will speak about the potential of immunization for sepsis prevention. Dear Stefan, the floor is yours. Thank you for being with us.
Thank you so much, Mariam, and hello, and thank you for having me here. Yes, I want to talk a little bit about immunization, vaccination, and its role and impact in sepsis. And to set the stage, let me just repeat what you know already. Sepsis is a major issue with almost 50 million cases annually, more than 13 million deaths annually. And in addition to sepsis, another problem, which is closely related, I want to mention today every now and then, is antimicrobial resistance, where about 4 million deaths annually are associated with AMR, and more than 1 million deaths annually attributed to AMR. And both are intertwined, and they're highly intertwined, and therefore can provide a perfect storm, which is much more threatening than either alone. Let me just mention briefly the successes of vaccines, and because they are very important, and I think we can get much more out of these vaccines for control of sepsis and AMR. In the United States, over the more or less last 30 years, childhood vaccination has prevented more than 1 million deaths. In the expanded program for immunization, mostly for newborn infants, small children in developing countries or low-income countries, over the last 50 years since its existence, 115 million deaths have prevented. And looking more at adolescents and adults, and perhaps the most impressive data are the immunization against COVID-19 in the European region. In only 28 months of being used and harnessed, 1.6 million lives have been saved. Globally, you can roughly estimate that every year more than 3 million deaths have been prevented by vaccines, and it is highly cost efficient. And for $1 spent, a return of $50 or more in direct and indirect costs can be expected. And there is a hidden bonus, and I want to stress the first bonus, and then comes the second bonus. Vaccines can also prevent emergence of antimicrobial resistance. That has been clearly shown with the introduction of pneumococcal vaccination, where in a high-income country, the United States, 64% decrease in the antibiotic resistance of the causative agent, obviously, for pneumococcal disease in children could be achieved, and about 45% decrease among adults. And also in a low middle income country, a member of the BRICS states, South Africa, an 82% decrease in antibiotic resistance among children has been observed due to the introduction of pneumococcal vaccination. And I want to stress that point and also go a little bit in more detail. Not a single pathogen ever has developed resistance against the vaccine. And that is in marked contrast to what happens with drugs, with antibiotics, because their resistance is increasingly disturbing and causing a lot of issues. Let me look at that a little bit in more depth. Let's first look at antimicrobial resistance, that is drug resistance in microorganisms, in pathogens. It happens when the drug target molecule, the molecule that's targeted by the drug, has changed by mutation and then selection of the new strain. And that excludes a whole class of antibiotics. It's not a single one, it's a whole class of antibiotics. And it requires a totally new class of antibiotics if you want to treat against this particular resistant pathogen. It occurs obviously in response to drug treatment within the patient or also outside of our body. For example, if the same drug family is being used for farmed animal farming and for industrialized animal farming. In some cases, these resistance can even be transmitted to other bacteria, meaning that microbes in your gut, your microbiome, may confer resistance to pathogens that enters the gut. And as a conclusion, we are really at the risk of running out of new drugs. A general solution would be that we find new classes of antibiotics which interfere with much more broader general features, mechanisms in the pathogen, rather than just by blocking a single key molecule, a target molecule. For example, by increasing permeability and disruption of bacterial cell walls or membrane. Now let's look at vaccine-induced immunity. Yes, evasion for vaccine immunity occurs, and we all are aware COVID-19, when the Omicron variant came up, the vaccines were less or not effective. But that is due to a single change in the protective antigen, the antigen that's recognized by the neutralizing antibodies which prevent infection or disease. So it's very, let's say, technically relatively simple to exchange the protective antigen because the construct, let's say a messenger RNA vaccine, remains and still works after modification. And that occurs independently from the immune response because mutation and selection is outside and also replacements can occur. So as a general solution, integration of conserved antigens which are recognized by antibodies would broaden and solve part of the problem, and that would be even stronger if the involvement of broader antibody functions and of T lymphocytes would be included. Now let me look a little bit at the vaccines as we have them in use currently from the side of the pathogen and the disease. And let me state it very clearly, for different pathogens, we don't have a single vaccine type. Rather, we need certain unique type of vaccine for almost each and every type of pathogen. First, we have the vaccines against acute infections. The example is COVID-19 or pneumococcal pneumonia, which can lead to sepsis. Single pathogen, a single antigen, and the vaccine is directed against that single antigen, and that's sufficient and efficient as long as it's stable. And neutralizing antibodies are directed at that single antigen, and they can protect from acute infection of the examples I given. It's getting much more complicated if the vaccine is against chronic infection, for example, tuberculosis. Here, a single pathogen expresses multiple antigens, and there is not a single protective antigen and neutralizing antibodies will be insufficient or don't work at all. Because the pathogen is hidden in cells, for example, a fine-tuned T cell balance is needed. So it's not just the T cell that shoots against the infected cell, rather it's a fine T cell balance, and therefore the antigen that's recognized by the T cells is also uncoupled from function, because the T cells see a diversity of antigens irrelevant from the function in pathogenicity. Vaccines against sepsis combine both. So we have vaccines that are needed for acute infections, and we have vaccines that are needed for chronic infections. So we have multiple pathogens, multiple antigens. In some cases, neutralizing antibodies work. In others, we need a fine-tuned T cell balance. So what are the vaccines that we have available? In principle, they come in two types: subunit vaccines or whole cell vaccines. Subunit vaccines are those which I mentioned already for the acute infections. They stimulate a neutralizing antibody, and the protective antigen is the antigen that induces neutralizing antibodies. That holds true for some acute viral infections, some chronic viral infections, for toxin producers where the antibodies just need to neutralize the toxin, and for encapsulated bacteria like pneumococcal pneumonia, where you can attack the capsule and therefore stop invasion and infection by the pathogen. And the vaccine types are either proteins plus adjuvants, carbohydrates, conjugates, viral or bacterial vectors expressing protective antigens, outer membrane vesicles, and messenger RNA enclosed in lipid nanoparticles. I don't have the time to go through these different vaccine types, but let me just come to one more point. Evasion occurs, and I mentioned it before, and therefore it occurs because evasion allows now evading the protective antigen or epitope. A second major factor where these subunit vaccines do not work if we don't have a protective antigen at all, as we have it in chronic infections, often like tuberculosis, where we need multiple antigens. And that is also due to the intracellular persistence. So induction of antibodies against conserved antigen epitopes under minimal or low immune pressure, that is conserved antigens, would improve such types of vaccines. Then we should have a stronger involvement of a whole spectrum of antibody functions, not just neutralization, but also antibody-dependent and cytotoxicity, complement activation, and others. And finally, T cell functions could also be involved and would improve vaccination against acute infections, because that is clearly something that can add to the neutralizing antibodies very relevantly. Let me just briefly mention not the different vaccine types, but the importance of adjuvants in protein antigen subunits and the importance of lipid nanoparticles for mRNA vaccines. Now we have more or less three types of adjuvants: aluminium salts that are well known, and they stimulate antibody production by B cells and help the antibody production. The more newer recent adjuvants are either emulsions or liposomes. Emulsions and liposomes in general have squalene, saponin, or other factors that allow the formation of water-oil formulations, and emulsions are much better in stimulating Th1 cells and cytolytic T cells, but not so much Th2 cells and antibodies. Liposomes, finally, they do both. They stimulate Th1 cells, cytolytic T cells, and also Th2 cells, which then can activate antibody-producing B cells. And the second advantage over the last years that came is the identification of pattern recognition receptor agonists, that are molecules that are recognized by our innate immune system in a very targeted and specific way. And one of the important pattern recognition receptor agonist is monophospholipid A, and that is involved in the adjuvants of the GlaxoSmithKline, but also some TLR7/8 agonists like imiquimod and resiquimod. So these are the two aspects, liposomes plus these agonists for TL receptors. Lipid nanoparticles are very important for improving or even making possible messenger RNA vaccines. They have a similar principle, but they are also nanoparticles, lipid particles. They are a little bit like liposomes. They contain cationic lipids and neutral lipids, which first stabilize the nucleic acids. They are here in the surrounding, but also the RNA inside is stabilized and protected. And importantly, they allow the escape of the nanoparticle into the cytosol, where the RNA then can be translated into proteins. Cholesterol is also integrated in these nanoparticles, and finally, polyethylene glycol constructs are sometimes, not always, part of these lipid nanoparticles. And I would dare to say that it is these nanoparticles that made a great part of the success of the RNA vaccines. The second group of vaccines are whole cell vaccines, and those come either in activated, killed forms. They are more or less a bunch of antigens and may need adjuvants. More importantly, in my opinion, in these whole cell vaccine antigens are the viable vaccines. They have inbuilt adjuvanticity. We know them from vaccinia against smallpox, BCG against tuberculosis, and there's a number of strong progress going on in further developing vaccines just a la carte, in a sense that they do what they do without major side effects. These vaccines are really needed if pathogenicity, virulence depends on multiple factors, not just attachment, as for the acute vaccines, as we see it with HIV for a chronic viral infection, or, for example, with tuberculosis or Staph aureus as a member of the sepsis-causing agents. In summary, I would say we have a plethora of pathogens that employ very different virulence and survival strategies in sepsis and antimicrobial resistance, and together there is a perfect storm. Complex and diverse host responses underlying sepsis AMR are important. We need to better understand those to get and develop further better vaccines. And different vaccine types are needed. It is not one fits all. Yes, R&D for vaccines against sepsis and AMR is complex and tedious, but new vaccines can markedly contribute to the reduction of sepsis and AMR. If I may, I have a very short outlook on the future of vaccine R&D. It includes both incremental and breakthrough research. First, we need vaccines which are rather technically easy to develop because it's a known area. For example, new vaccines against replacements of Streptococcus pneumonia. We can use our known strategies to target unknown pathogens for which we have no vaccine yet, unknown vaccines against those, like Klebsiella pneumonia, and again, similar vaccines could work. In the area of unknown known, it's very important to develop novel vaccine types, and notably to get universal vaccines that will combat also different types of one of the same pathogen. And finally, breakthrough research ultimately will need it, because ultimately, with the means of artificial intelligence, we want to develop precision medicines for preventives and for therapeutics. And I think that will be the final step to prevent and also therapy and also drug treat with such precision immune-based intervention measures. And I think I have to close now, and thank you so much for your attention.
Stefan, thank you so much for the comprehensive presentation, as always, and the audience were delighted to listen to your speech. We are compiling the questions and answers. Most of the comments were just positive feedback and welcoming, and we will come back to you with the questions. As the conclusion to your presentation, let me highlight that the 2030 Global Agenda for Sepsis, that is our very first multi-year global strategy, clearly emphasizes the importance of immunization agenda, both at the community level and through strengthening national immunization programs at the healthcare facilities. And until we have the new and novel vaccines that could be effective against bleeding pathogens, we definitely have to prioritize strengthening of the routine catch-up or campaign immunization against the most vulnerable and high-risk groups. Thank you very much, Stefan.
Thank you. Thank you, everybody.
Thank you. Let me move to our second presenter. Professor Shabir Mahdi is a prominent South African vaccinologist and infectious disease expert, renowned for his pivotal contributions to global health. Currently, he is the Dean of the Faculty of Health Sciences at the University of the Witwatersrand in South Africa. As a professor of vaccinology and director of the South African Medical Research Council, he has led groundbreaking research on vaccines against pneumococcal, rotavirus, and COVID-19, among others. His work has significantly impacted public health policies, especially in low and middle income countries that bear the largest global burden of sepsis. Professor Mahdi will present on maternal immunization to prevent neonatal infections with sepsis agents. Professor Mahdi, the floor is yours.
Thank you, Mariam, and thank you for this invite. So just before I start, a few declarations of interest, much of which the funding goes to the institution that I work at. So what I'm going to do over the course of the next 10 minutes is firstly just reflect on some of the recent advances in the prevention of neonatal sepsis through maternal immunization, and then delve into some of the opportunities and challenges that we're likely to face in terms of prevention of neonatal sepsis through maternal immunization. So when we think of maternal immunization, I think it's important to reflect that this is a program that dates back to almost 150 years ago when smallpox vaccine was recommended for pregnant women because of the high burden of smallpox in very young infants. And over time, there's been a number of efforts in terms of protecting young infants by vaccinating their mothers during pregnancy. Currently, perhaps the flagship program in terms of the impact of maternal immunization has on neonatal morbidity and mortality really speaks to the issue of the tetanus vaccine, where since its recommendation in the late 1980s, it's attributed to save more than 750,000 lives, together with improved care of the cord amongst newborns. The current recommendations by the WHO is for all pregnant women to receive tetanus toxoid vaccine. In addition, WHO also recommends that women receive influenza and COVID-19 vaccines. Both of those vaccines have been shown to be effective in preventing illness in the mothers during the first 3 to 6 months of life, in addition to which those vaccines are also attributed to reducing preterm birth as well as stillbirth amongst vaccine recipients. Most recently, WHO has now made a recommendation for pregnant women to receive RSV vaccine during each of their pregnancies. And I'm going to speak a little more with regard to the impact that the RSV vaccine is expected to have in terms of neonatal infections and how that might also have some sort of positive spill in terms of reducing emergence of antimicrobial resistance. And then I'm going to touch briefly in terms of further developments around a group B strep vaccine. And again, the GBS vaccine, the efforts of developing a vaccine against group B strep dates back as much as to the late 1970s. And it is only more recently that we probably have a vaccine that is likely to go into clinical trials in phase 3. So when we think of RSV, it's about 100% of children will be infected with RSV at least once in the first 2 years of life. Importantly, each year there's about 33 million episodes of RSV LRTI, of which 10% result in hospitalization. And an estimated 100,000 babies would die of RSV each year, with 97% of those deaths occurring in low middle income countries. roughly about 45% of all of the hospitalization and deaths due to RSV occurs in children under 6 months of age. So these are data which are probably familiar to most people, and these were the results of the first vaccine that has been licensed for pregnant women to protect infants against RSV illness. And what is observed in these studies is that both in high as well as low middle income countries, the vaccine efficacy against severe medically attended LRTI was 70% overall, with not much difference between high compared to the low middle income countries. In addition to which, vaccine efficacy against medically attended LRTI through to 6 months of age was roughly about 50%. Now, this vaccine perhaps is more immunogenic and more efficacious than an earlier nanoparticle vaccine that was evaluated also in pregnant women. Unfortunately, for this particular vaccine, it didn't meet the primary endpoint for licensure. But one of the important findings of this earlier vaccine of RSV in pregnant women was a reduction in terms of antibiotic usage among infants born to women that received the RSV vaccine. When looking at the overall analysis, there was a 13% reduction in antimicrobial prescriptions to the infants born to mothers who received the vaccine, and 16% reduction for antimicrobial prescriptions in relation to LRTI episodes. Strikingly, in high-income countries, there was almost a 50% reduction in antimicrobial prescriptions for LRTI. So in addition to preventing a viral infection, because of the largely indiscriminate use of antibiotics to treat mild infections in infants in early childhood, there is a potential for these vaccines targeted at viral pathogens also bring about a reduction in terms of antimicrobial usage. Now, in addition to that, the other pathogen for which a vaccine is now nearing into phase 3 trials is vaccines against group B strep. GBS is estimated to cause roughly about 320,000 invasive episodes in infants, primarily in the first 3 months of life, but about an estimated 190,000 infants dying each year. And the majority of those deaths also occur in low and middle income countries. In addition to which, GBS in pregnant women is also estimated to conservatively cause at least 57,000 stillbirths, as well as up to 3.5 million preterm births. So these are data that were published recently in terms of the immunogenicity of the vaccine. And as is evident, the antibody concentrations in babies born to women that received the vaccine was substantially higher compared to that what was observed in babies born to placebo recipients. One of the important features in terms of vaccines that are targeting the capsule of the different bacteria, including GBS and as I'll allude to when it comes to Klebsiella pneumonia, is that there seems to be some sort of heterogeneity in terms of the efficiency of transplacental antibody transfer. And to some extent, the transplacental antibody transfer is less efficient in low middle-income countries compared with what is observed in high-income countries. Now, in the same study, what was evaluated was in parallel to the immunogenicity and safety study, we also undertook a study to identify whether we were able to identify a threshold associated with risk reduction of invasive disease. And the reason for this is that undertaking an efficacy trial for GBS vaccine in pregnant women would entail needing to enroll over 60,000 pregnant women, which would make it a really difficult ask in terms of getting to licensure. So in this cohort study, we were able to identify a threshold associated with 80, 90, 95% risk reduction of invasive disease. And looking at immune responses and antibody concentrations in the infants, we were then able to estimate that this hexavalent GBS vaccine would probably have an efficacy, predicted efficacy of roughly about 83%, ranging from 65% to 96% at the individual serotype level. So the agenda here is really to try to look towards getting licensure of the GBS vaccine based on establishing safety in a pregnant woman, as well as benchmarking it against this correlate of protection or threshold associated with risk reduction. I'm wanting to now really conclude in terms of what are the further opportunities and challenges for prevention of neonatal sepsis through maternal immunization. Now, when looking at, this is the data from what is known as the Child Health and Mortality Prevention Surveillance Program, which is being undertaken in 9 different low and middle income countries, mainly on the African continent, but also 2 in South Asia. And when looking at post-mortem investigation as to the cause of death in these babies, what is quite evident is that in a neonatal period, close on to 45% of neonates that die have got an infectious etiology somewhere in the causal pathway. In a large percentage of those deaths where infection is attributed in the causal pathway to death, it is not an underlying cause of death, but rather an immediate or antecedent cause of death. When looking at those cases where infections are attributed in the causal pathway to death, what is quite striking is that irrespective of at what age the death occurred during the neonatal period, be it less than 24 hours, 1 to 2 days, 3 to 6 days, or 7 to 27 days, is an eminent role of gram-negative bacteria, including Klebsiella pneumonia. In babies that die within the first 24 hours of life, we also got a reasonable percentage of those infection-related deaths being attributed to group B strep. But beyond 24 hours is really the dominance of Klebsiella pneumonia, A. baumannii, as well as E. coli. Now, looking at it across the different pathogens, and these are specifically for deaths that are attributed to community-acquired infections, we find that Klebs pneumonia is responsible for 30% of the community-acquired infection-related deaths overall, with some sort of heterogeneity across the different countries, ranging from about 10% in South Africa to close on to 60% in Mozambique. In addition to which, what we also find that in a country such as Bangladesh, although A. baumannii is considered to be largely a hospital-acquired infection, we find that in Bangladesh it being reported as a community-acquired infection and something which needs to be delved into further. When looking at presumed hospital-acquired infections, again, the eminent role of both Klebsiella pneumonia and A. baumannii as the leading pathogens associated with the presumed hospital-acquired infection-related deaths. And what we find is that of those pathogens attributed to having acquired in hospital that has led to death, about 50% of those are due to Klebs pneumonia overall, and roughly about 35 to 40% due to A. baumannii. When looking at the country's sort of breakdown, that ranges anything from roughly about 30% to up to about 85% in Ethiopia being attributed to Klebs pneumonia. Another important feature of this particular study is that the majority of these presumed hospital-acquired related deaths are occurring in preterm births, and that is important in a context of maternal immunization, as I'll show you just now. Now, another striking feature of this Klebs pneumonia that we observe as being the dominant cause of neonatal deaths associated with infections, both in the community as well as the hospital-acquired infections, is a diversity of sequence types as well as the diversity of K antigens that are involved, that are identified in those isolates. And notably, it isn't the same K antigen which is dominant which is dominant across all of the different sites, with up to about 80 different K antigens are being identified in the study. In addition to which, although there are more limited number of O antigens for Klebs pneumonia, we do find that there's some level of heterogeneity in terms of which O loci are identified in these invasive Klebs isolates. Now, in addition to the heterogeneity of the K antigen types that is identified across the different sites, I think what's also important to recognize in the context of Klebs pneumonia is the temporal changes that occur over time. And these are data from Malawi as an example, which shows that the sequence type as well as the K types, as would be the case, does actually vary, which makes it really difficult in terms of determining which antigens to include in Klebs pneumonia vaccine. So in conclusion, in terms of the challenges that we face, Klebs pneumonia is clearly a leading cause of community-acquired and hospital-acquired sepsis amongst neonates. But we do face a challenge that there is temporal as well as geographic heterogeneity in the circulating strains of Klebs pneumonia, which has got implications as to which K antigens would need to be included if the K antigen included as the target in a vaccine. As I indicated, a high percentage of these deaths do occur in preterm births, which could diminish the potential for maternal immunization as a strategy, because the timing of the vaccine during pregnancy would need to be relatively early, and because of the low efficiency of transplacental antibody transfer, that would result in babies that are born at preterm. So another option to consider in terms of reducing neonatal sepsis, over and above obviously infection control practices, is a potential for a cocktail of monoclonal antibodies to neonates that are at high risk for hospital-acquired infections. Thank you for your attention.
Thank you so much, Professor Mahdi. You have shared some of the potentially groundbreaking research findings that can contribute not only to the global knowledge basis, but specifically breach the data gaps and evidence, the scarcity of evidence from the low and middle income context, and especially from community onset sepsis cases. And we look forward to the publication and further discussions of the evidence, both in terms of RSV and GBS vaccination potentials for reducing the neonatal sepsis incidence and mortality. Thank you very much. We have only one question currently in the chat, but we will look for other questions and have the Q&A session later on. Therefore, it's my pleasure to move to our next presenter, Professor Elie Saade, who with over 15 years of experience in infectious diseases, geriatric medicine, and public health, focuses on clinical excellence, research innovation, and academic leadership. As Medical Director of Infection Prevention and Control, he leads initiatives to enhance patient safety. His research expertise includes bacterial infections, particularly extraintestinal pathogenic E. coli, antimicrobial resistance, and healthcare-associated infections. Elie has led over 15 million in grants from CDC, National Institutes of Health, and industry partners, advancing infection prevention strategies and vaccine efficacy. We will have the opportunity to listen to his experience and prospects on the novel vaccines for sepsis prevention. Professor Saade.
Thank you, Doctor. Hi, everyone. I'm Elie Saade. I'm calling from Cleveland, Ohio today. It's snowing now, but not too much, so we're good. So I'm going to be talking about the vaccines for sepsis prevention. I understand, and I attended the previous talks, and they were very interesting. I will not be able to top any of that, but I will try to give an introduction to current existing vaccine, as well as some vaccines that are being studied. It's just one first disclosure here is that my experience is mainly related to the clinical trial phase, and it's mainly related to respiratory viruses, as well as contributing to clinical trials for some bacterial vaccines. So these are my conflict of interest, and I will be mentioning some of these products. All the information that I will share is publicly available, and it only represents my opinion and not that of any of these companies or other entities or my school or my hospital. So after the introduction, I'll be talking first about vaccines for the top 15 causes for sepsis worldwide. Then I will be going into a little bit of details for vaccines targeting E. coli, Staph aureus, Klebsiella, pneumonia, Pseudomonas, and a general vaccine against antimicrobial resistance. So these are the top 15 causes of infection, or actually 16, for sepsis worldwide, not necessarily in orders. So I'm sure a lot of those were already mentioned in previous talks. As we can see, we're talking about, so for every vaccine here is divided between preclinical phase 2, phase 1 or 2, and then phase 3, and then approved. So one thing that we can note, just looking at this in general, is for the approved vaccines are very limited. So we have vaccines that are approved for strep pneumonia, and as with everything here, when it rains, it pours. It means like there's not only one vaccine for strep pneumo. There's many vaccines that are available that are the subjects of other talks here that I'm not going to be discussing. Then we have the vaccines against Salmonella typhi, both Salmonella typhoidal and non-typhoidal Salmonella. Some of those are already approved and available clinically, either in the US or elsewhere. There's Haemophilus influenza B vaccines. We have the meningitis vaccines. And BCG is also mentioned here, even though BCG vaccines have a lot of problems. Otherwise, the phase 3 trials will, like most vaccines in the phase 3 trials, are either discontinued or failed already. There's still some active trials ongoing, especially for vaccine, especially for diseases where we already have vaccines available. And I'm going to be talking about it in a minute. So phase 1 and 2 and preclinical, we have a lot of candidates, but as we all know, all these candidates might fall off at some phases here, unfortunately. So this is the vaccines or the organisms that have vaccines already available and that can be given to patients. And these are the ones with the phase 3 clinical trials, with the caveats here that we're showing some of them which are already discontinued. So there's no ongoing clinical trial for Staph aureus that I know of. So expect extraintestinal pathogenic E. coli is actually 9. It's not 10, but it's also discontinued. And I'll be talking about it a little bit. There's Pseudomonas vaccines failed, C. diff vaccines failed, and so forth, unfortunately. So starting by vaccines targeting E. coli. E. coli and Staph aureus are both on the top of the list for sepsis wide. The main target for E. coli would be the extraintestinal pathogenic E. coli, which is the strains that cause invasive bloodstream as well as invasive sepsis and invasive infections related to urinary tract infections or other sources as well. Is a leading cause of adult sepsis and top cause of urinary tract infection worldwide. It's commonly isolated in patients with urosepsis, intra-abdominal sepsis, and bloodstream infections. So there's a very high burden of disease and lack of effective prophylactic measures with the increase of antimicrobial resistance and other factors, which sort of drives the need for a vaccine to prevent invasive E. coli. So expect, again, 9V. So the 9-valent extraintestinal pathogenic E. coli vaccine. It's a polysaccharide conjugate vaccine targeting the most common serotypes. It's a vaccine that showed robust antibody responses in older adults in phase 2 trials. In phase 3 trials was recently, which was ongoing in patients who are older than 60 at high risk of urinary tract infections and sepsis. The study, unfortunately, was discontinued early this year in February, as was announced by the company working on this. It wasn't because of safety issue, it was because of an interim analysis where it failed to meet efficacy endpoints. So it was discontinued because of futility, unfortunately. So that was lost. There's that was lost so far. Again, I only have public information. So other candidates, so there's the protein subunit vaccines targeting adhesion proteins or iron uptake system. And there's also oral immunotherapy, which have variable efficacy, not really well demonstrated or formally used. So currently, there's no E. coli vaccine approved for human use. Failure of the phase 3 highlights the challenges due to heterogeneity of the E. coli strains. Future research aims to refine the targets, looking at conserved proteins. and combined approaches to improve efficacy. Next, I'm going to talk about vaccines targeting Staph aureus. Staph aureus is a leading cause of sepsis and septic shock globally, commonly implicated in bloodstream infections from skin and soft tissue, and pneumonia and device-related infections. It's the single deadliest bacterial pathogen worldwide, associated with 1.1 million deaths, approximately a year. This number is from 2019. It's highly virulent due to its ability to produce toxins and evade immune defenses. There's like a big number of vaccines that have failed in phase 2 or phase 3 trials over the past few decades. So some candidates that failed or stalled: Stapvax, which is a polysaccharide conjugate that includes capsular types 5 and 8. It failed in phase 3. Another candidate is a single antigen vaccine that was also failed in phase 3 because of efficacy and safety concern. Another tetravalent vaccine that includes capsular polysaccharides and toxins, which from what I could tell is still in the pipeline. Currently, researchers are focusing on multi-component vaccines combining surface antigens and toxoids. So again, a combined approach to overcome Staph aureus immune evasion. Successful vaccine could significantly reduce invasive staph infection and sepsis, especially in high-risk groups like surgery patient post-surgery, dialysis patient, so such that the heptavalent toxoid vaccine targeting 7 exotoxins now in early clinical development. The LBT-SA7 multivalent protein vaccine, and the 5 antigen vaccine from JSK, still in phase 1 trials. Vaccines targeting Klebsiella pneumoniae. So Klebsiella is a common cause of hospital-acquired pneumonia, urinary tract infections, intra-abdominal infections, progressing to sepsis. It's among the top 5 most lethal bacterial sepsis pathogens, globally notable for multi-drug resistant infections, such as carbapenem-resistant strains, which are a WHO critical priority. And there are hypervirulent strains that can cause community onset sepsis, such as liver abscess with bacteremia. So current vaccine development. So there's the capsular polysaccharide vaccine. It's an early 24-valent vaccine tested, not commercialized. It's there's early clinical trials ongoing. Lipopolysaccharides or antigen vaccines targeting fewer antigens, also in preclinical stages. Protein subunit vaccines targeting conserved protein, focusing on enhanced immune response, and whole cell and OMV vaccines, which can provide experimentally broad coverage approaches, including vaccines that show protection in animal models. And so this is a recurring theme in my talk, which is a little bit depressing. Again, so a lot of challenges related to the high capsular diversity, more than 70 serotypes, multivalent conjugate vaccines combining capsules or antigen and protein subunits might be promising. The focusing on high-risk groups, especially in the initial stages. E. coli is definitely, Klebsiella is definitely a problem because of the microbial resistance and increasing incidence. So that's a challenge to keep in mind. Vaccines targeting Pseudomonas aeruginosa. So it's a highly virulent, opportunistic pathogen causing nosocomial sepsis, commonly associated with ventilator-associated pneumonia, wound infections, and bacteremia in ICU patients. It's among the top 5 pathogen, again, causing sepsis death globally. Frequently multi-drug resistance and also designated as a critical priority. It's very fatal, especially in immunocompromised patients, where the mortality can be 50% or more. So some of the vaccine candidates, so there's the octovalent or polysaccharide conjugate, which got to phase 3 in cystic fibrosis patient, but failed to prevent chronic lung infections. The bivalent flagella vaccine targeting major flagellar types A and B, also lack of efficacy in cystic fibrosis. The IC43 or OPRF fusion protein, phase 2 showed good safety and immunogenicity, and slight but not significant mortality reduction. And there was no phase 3, no significant reduction in pneumonia or mortality. So some of the future directions, mRNA vaccines and vaccines targeting type 3 secretion system components. These are in early clinical trials, both types. The whole cell or the outer membrane vesicles vaccines, the monoclonal antibodies, which is passive immunization, which can have some synergy when combined with vaccines. The future direction would be to combine multiple antigen for broad protection and also prioritization of high-risk groups. A little bit of change in theme here, talking about vaccines against antimicrobial resistance and not specific organisms. So those are vaccines that are conceptually would neutralize resistant mechanisms to restore antibiotic efficacy, targeting the beta-lactamase, the efflux pump proteins, resistant plasmid factors, and modified drug targets like the penicillin binding protein 2A. Rationale, by raising antibodies against resistance determinant, we can prevent or reverse antibiotic inactivation. Most of them are currently preclinical. There's promising data from animal models and in silico design. Vaccines targeting beta-lactamase and efflux pump. So beta-lactamase, so the NDM1 and the MC subunit vaccines showed neutralizing antibodies that restored beta-lactam antibiotic activity in preclinical study. The challenge is that many beta-lactamase, as you know, are periplasmic, limiting antibody access, and also these are very diverse enzymes. The efflux pump vaccines targeting outer membrane components with enhanced opsonization and improved survival in resistant infections. Efflux pumps antigen are surface exposed and conserved, so they're easy to access in this process. But the protection can be partial, which will require multiple antigen approaches. There's vaccines targeting plasmid proteins, like vaccines against surface exposed conjugative proteins, which can protect against bacteria carrying the plasmid by inducing antibodies that will bind the plasmid encoded protein, preventing transfer, helping clear plasmid-bearing pathogens. Vaccines targeting the PBP2A of the MRSA is neutralizing the PBP2A, which will increase the beta-lactam susceptibility and improve bacterial clearance. The future direction will be combining multiple antigens to broaden coverage and develop outer membrane vesicle-based platforms for hidden intracellular targets. Again, going back to our table, a lot of promises. So far, very few are kept, except for pneumonia, meningitis, and a few others. Thank you. And these are some of my references, and there's a lot more but request.
Thank you, Elie. Such an insightful presentation, and the latest data on initially promising, but lately we learned that some of the advanced clinical trials had to be discontinued for a number of vaccine candidates. And this, again, is the stronger reason and rationale for our continued advocacy for increased investments in sepsis research, especially in prevention commodities, including new vaccines. And I believe that the Global Sepsis Innovations platform that we recently launched, based on the US Sepsis Alliance experience and FDA-facilitated process for sepsis innovations, we believe that we can join the voice and consolidate our positions to advocate both for increased investments and for consolidating evidence and approaches in collaboration with BARTA, HERA, other relevant authorities and regulatory authorities, as well as the SAGE of WHO. So, and of course, with the high-risk groups, including neonatal or older adults or immunocompromised individuals, we definitely know where the research priorities should be, but we need continuous support to the private sector and the vaccine industry to incentivize finding new prevention solutions. Thank you very much. And let me introduce our final speaker, Dr. Jeremy Brown, who is the academic respiratory consultant, leading a team at University College London on investigating the pathogenesis and mechanisms of immunity for pneumonia. He also conducts translational research into patients with bronchiectasis, pneumonia, and COVID. Dr. Brown is the co-lead of the UCL Respiratory Department, leading a group of 10 investigators with research groups on a range of lung conditions. Since 2018, he has been a member of the UK's Joint Committee on Vaccination and Immunization, contributing to national decisions on vaccine policy for multiple vaccine-preventable pathogens. And the presentation will focus on vaccines for hospital-acquired infections. Jeremy, the floor is yours.
Actually, Elie has very helpfully set the scene for my talk. I'm going to talk about vaccine approaches, well, the possibility of getting vaccines for hospital-acquired pathogens. And this is a list of hospital-acquired pathogens in here. I start with the viruses because actually in many hospitals, there's quite a lot of infection that occurs with viruses between patients. And really, we do have vaccines for those, such as influenza, RSV now, and COVID. And they are really very important for reducing hospital-acquired infections. But there's a whole bunch of bacteria, and Eli's mentioned several of them: Staph aureus, Pseudomonas, E. coli, C. difficile, Klebsiella pneumoniae. These are the very common pathogens which cause infections in the hospital. And we do not have any vaccines for any of them as yet. This is a major issue in clinical practice because we cannot prevent the common causes of bacterial death in hospital. This is a World Health Organization report from 2021, which gives a list of vaccine development for bacterial pathogens. You can see this on the left-hand side. There's a table. There's a whole bunch of vaccines being developed for pneumococcus, which is a little bit ironic because we have a very good vaccine already. But pharma, when they identify something where you can make a profit, will try and do Me Too vaccines, which is what is happening in pneumococcus. And the pathogens that we're interested in, E. coli, Klebsiella, Staph aureus, C. difficile, various Enterobacter and Acinetobacter and Pseudomonas. We don't have vaccines in active clinical development. And over on the right-hand side, this is illustrated even more with the phase 1, phase 2, phase 3 trials, and the actual number of vaccines being introduced is really very limited. So we have a big issue. We have a really big issue in that the vaccine development is not happening for hospital-acquired infections, whereas it is happening for community-acquired infections to a certain extent at least. This is an illustrative example, but in fact, Eli's whole talk was an illustrative example of how things happen in vaccine development for bacterial pathogens, which is that you identify multiple different potential vaccine approaches. There's a lot of preclinical work, and some of those reach clinical work. And so, for example, in Pseudomonas, there's been at least 100 plus preclinical candidates, 10 have reached clinical trials, and none have come into clinical practice. And the main reason why is lack of efficacy, sometimes toxicity for LPS type vaccines, but it's largely failure of efficacy. So there's a big issue. And it's not easy to develop a vaccine against hospital-acquired infections, it seems. And why is that? So there are several very good reasons why it's quite hard to get a vaccine for a hospital pathogen. The first is that many of these pathogens are genetically highly varied. They only share about 50% of their genes with all the other strains of, there's only 50% of the genes which are common to all strains of these pathogens in general. So this is Acinetobacter baumannii. I do some work on Acinetobacter baumannii, which is why I've got this picture here. And you can see that there's a very large number of genes have been identified. Each genome itself is about 4,000 genes, but the actual number of conserved genes is going down to about 2,000, 2,500. So the actual potential protein targets is relatively limited, which are shared across all strains. And each of those targets has to be surface expressed, it has to stimulate an immune response, and it has to be highly expressed during infection as a target for a vaccine. And whatever you induce with the vaccine, whether it be IgG responses or cellular responses, when it recognizes that antigen, it has to induce a protective response. It has to boost immunity, and it doesn't necessarily do that. You can have many antibodies which will bind an antigen, but do not actually improve immunity. Also, Eli touched on this as well, is that you have the problem with the capsule. So many of these pathogens are surrounded by a capsule. The capsule blocks access to the proteins, which are the conserved antigens beneath the capsule. You can see that on the flow cytometry picture. So this is Acinetobacter with a capsule and antibodies to two different proteins. You get a bit of a shift to the right in the bacteria. Take the capsule off, you get a big shift. So the binding is much stronger. So although you can get a response which will work through a capsule against conserved proteins beneath the capsule, it is much weaker than it would be against unencapsulated organisms. And targeting the capsule itself, you might ask, is a good idea. That's what we do for pneumococcus, what we do for Haemophilus. However, often the capsule structure varies markedly between strains. So, for example, there are 30 different capsule structures. Common Acinetobacter cause infection commonly with Acinetobacter, and Klebsiella is similar. So these are two sort of biological reasons why there might be major issues with trying to develop a vaccine. And a third one, which is probably underappreciated, is that these pathogens, many of them are commensals, or we have been exposed to frequently. So the target population, humans, have an adaptive immune response to many of these pathogens already. So what you're trying to do with a vaccine is overcome a weakness in your pre-existing adaptive immune response. And that is a much harder thing to do than to boost somebody from no adaptive immune response to an adaptive immune response that has some protective value. So to improve something which is already existing is a harder thing to do than to make something de novo. Also, these organisms cause infections at different sites. So you can get Klebsiella pneumoniae causing pneumonia, cause septicemia, and it can cause a UTI. Each of those sites, the protective immunity you may require might vary. Antibodies clearly very helpful for protection against sepsis, but against lung infection, we're not so sure. The antibody is effective, but probably there's some component of cellular immunity will be beneficial as well. And compounding that is that it's actually harder to prevent a organ-specific infection than blood infection. And we know this from the pneumococcal world, where the pneumococcal vaccine is really very good at preventing sepsis or meningitis caused by a vaccine serotype. 80 plus, maybe 90% of it effective. However, it's only 50% effective against vaccine types causing pneumonia. It's harder for a vaccine to cause a protective immunity within an organ than it is within the blood. And then the pre-existing immune response can cause problems. There's the issue about trying to boost the pre-existing immune response, but there's also things like antigenic sin, which means sometimes, and this is the problem with the Staph aureus vaccine that was mentioned in the previous talk, the protein vaccine, is that actually when you vaccinate with that protein, all that happens is that you boost non-protective antibody, and that causes a failure of the vaccine, essentially. And there is always this potential for immune-mediated toxicity if you have pre-existing immune responses. And I worry about that for cellular immunity specifically. So these are technical challenges. And as Mariam mentioned, what we need essentially is more money. It is a bit of a Cinderella area, developing vaccines for hospital-acquired infections. And without a considerably more investment to really get to the grips of this, I don't think we're likely to succeed that easily. Once we have a vaccine, there are a few other issues. One is that the target population are the specific target populations that are the worst at responding to a vaccine. They're going to be elderly, and they're going to have some degree of immunosuppression often, the patients in hospital, due to their comorbidities. And you can see this here with the responses to pneumococcal vaccine. These are rheumatoid arthritis patients. Those on rituximab, that's a B-cell depletion therapy, so it's not unexpected that it affects vaccine responses. But also those on methotrexate, which is a standard treatment, and anti-TNF therapy, they also have poor responses to vaccines. Now, fortunately, in some ways, one good outcome from the COVID vaccination and the pandemic is that we do now have very good data on what treatments and what diseases really do affect vaccine responses. So when it comes to the point where we have vaccines to use, we now know which patients they will work the worst in and the patients they work the best in, and be able to counter that. Normally, with these vaccines, even in the immunosuppressed, there are some that will respond. And even if you have a poor immune response to a vaccine, it's often adequate. I mean, the clearest example of that is COVID vaccination. In the elderly, we know their antibody responses are weaker to COVID vaccination, yet You don't see severe COVID affecting the elderly very often now because they have all been vaccinated. And although it's a weaker antibody response than young people, it's still easily enough to prevent severe infection. There are other approaches as well. The data showing that if you stop methotrexate, vaccinate, and then restart methotrexate, then you're going to get an improved vaccine response. So you can overcome these issues that you might get with poor immunosuppression, with poor vaccine responses in the immunosuppressed and the elderly. Another major issue is that for hospital-acquired infection, I mean, if you're going to vaccinate, it's generally speaking, it's a vaccine is active. You have to vaccinate in anticipation of the disease. And that means for hospital-acquired infections, you have to anticipate who will be in hospital. And although that's relatively easy, they're going to have comorbidities and they're going to be old, or they may be coming in for elective admission for some surgery. It's still not that easy. And the risk population is quite broad. And that means there's a cost efficacy issue because you have to vaccinate a lot of people to prevent one potential case of hospital-acquired infection. That does make it a much harder process than, say, routine vaccination for RSV in children, where we know, in fact, that's going to be incredibly cost effective because 20% of children will get RSV and severely in their first year of life. There is an alternative, and that's been mentioned. That's monoclonal antibodies, passive vaccination, which you could use in really high-risk people in hospital, such as those in intensive care. And then the last issue is the potential of vaccine fatigue, because we're talking about a lot of pathogens here. So in the UK, the elderly get 4 vaccines routinely, and RSV is coming in, so that will be a 5th. And then if we add in some of the common bacterial and pathogens, Staph aureus, Clostridium difficile, E. coli, you can make a very good case for universal vaccination of the elderly for those 3 pathogens. And potentially you can target vaccinations for high-risk groups, Pseudomonas for people with chronic lung disease, for example, and so on and so on. You can end up having to vaccinate people for an awful lot of pathogens. And that becomes an issue because of acceptability and also cost efficacy. And this is something we have to, I mean, unfortunately, we're not in a position where this is an issue, but this could potentially be an issue in the future. So to conclude, we really do need vaccines against hospital-acquired bacterial pathogens, but we do not have them. And there are really very limited number of candidates in clinical development, and the history so far has been a failure, unfortunately. There are major issues of selecting which antigen to go for, the type of protective immunity that we need to prevent different types of infection, and how to protect against multiple sites of infection with one vaccine. And then there's pre-existing immune problem that we have, which is something that probably underpins many of the reasons why the vaccinations in the previous clinical trials have not worked well. And then once we have a vaccine, we do have the practical issues of making sure it works well in our target population whose immune system does not induce very good vaccine responses, and also identifying who to vaccinate in anticipation that they end up in hospital and at risk of infection. And all this is going to require good cost efficacy calculations, and that needs really good data on exactly how the infections are affecting different populations of patients in hospital. And overall, the big thing is we need more money, lots more money to develop this. Thank you for listening today.
Thank you, Jeremy. What a wonderful conclusion of our session, diving into the challenges of the hospital, of the vaccine against the hospital-acquired infections. And indeed, despite the burden of sepsis, both in community and hospitals, it's, of course, unacceptable that there is no vaccine development against the HAIs, despite due to the failure of understanding the immune requirements or a failure of advocacy. And despite the latest developments in the global health arena, especially against COVID vaccination and some of the cost-effective global health interventions, we shall continue to hope that those decisions will be temporary, and we will definitely witness increasing support to the life-saving vaccine and other prevention measures in the global health. And in this aspect, I believe that we have to join our advocacy efforts and maybe develop joint statements, official statements on behalf of the Global Sepsis Innovations Platform or other relevant platforms, and work even closer with WHO, Gavi, or the Medicines Patent Pool and other initiatives that can make both resources available and reach the decision makers that immunization, the most cost-effective and the biggest success story in the history of medicine and global health, should be not discontinued, but on the contrary, strengthened for new innovations and technologies and solutions. As we move into the Q&A session, again, looking through the chat box, we see a lot of notes of gratitude to the excellent presentations and congratulations on the findings from Dr. Mahdi's study, the maternal vaccination for neonatal sepsis prevention. And we have only one specific question from Ghana. So to what extent will population genetic polymorphisms like heterogeneity present challenges to development of vaccines to emerging and re-emerging infectious diseases, especially from pathogens which are also fast mutating?
Well, if you're talking about the pathogens evolving away from the vaccine, well, we sort of, well, there's a pneumococcal experience where you vaccinate against a certain number of serotypes and they are removed from circulation and other serotypes move in. I think that's going to be much less of an issue for hospital-acquired pathogens. The reason why it happens in pneumococcus is children are the reservoir of infection for adults as well. And hospital-acquired infection, I can't see that being such an issue. That's not really evolution in the form of the antigen being sort of removed. It's just a sort of what's the word? It's environmental pressure essentially from the immune system. I'm not sure because it's a bit confused, the question to me. I can't quite grapple with it. And then I think if you're talking about the subjects, i.e. the population who've been vaccinated with genetic polymorphisms affects the vaccine development, I don't think it does necessarily. I'm trying to think of an example where that may have been an issue. I think there's more an issue about their exposure to pathogens and how that might affect their response to a vaccine. Might be more of an issue because there may be different exposures in different populations. So sorry, not a very good answer to the question, I'm afraid.
I believe this question also resonates for the challenges of heterogeneity of etiopathogenesis and the need for multi-component vaccine candidates that you and Elie have also touched upon in your presentations. I would like to once again thank the audience. We're delighted to cross the 10,500 registrations for the current Congress. I would like to thank the amazing panel of our speakers. Gentlemen, it has been an honor and pleasure to moderate this session. And we look forward to our closer collaboration, the Global Sepsis Alliance, through our European Sepsis Alliance and African Sepsis Alliance branches. We will be in touch for your potential interest to join our latest initiative for the Global Sepsis Innovations Platform that is intended to bring together innovators, researchers, sepsis survivors, and policymakers for discussing the unmet needs in research and development for sepsis, and also discussing recommendations for advocacy or regulatory considerations. Thank you very much once again. And with this, we conclude the session. Also, we use the opportunity to let you know that starting from April 15th, we will start releasing the video recordings of each scientific session of the 5th World Sepsis Congress on the YouTube channel as well as podcast. We also use the opportunity to ask you to visit the Global Sepsis Alliance site and to endorse the World Sepsis Declaration as well as the 2030 Global Agenda for Sepsis. You can also follow us on the following social channels: Facebook, X, TikTok, as well as LinkedIn and Instagram, of course. And finally, we would like to thank our sponsors, Sanofi, bioMérieux, 144, Adrenamed, and Thermo Fisher. Without their support, convening the 5th World Sepsis Congress would not be feasible. And again, we look forward to strengthening public-private partnerships and investments in sepsis science for finding better and more cost-effective and affordable solutions. On behalf of the Global Sepsis Alliance, we thank you for your attention and being with us in this late hour.